So our next speaker is Antonio Lascano. He's a very good friend. We were talking yesterday about 25 years of friendship, so it's a long way we've come together. He's an evolutionary biologist. He's very much interested in the origin of life and the early evolution of life. And he has contributed many research papers to the scientific community, many outreach papers, also about 70 outreach papers, and several very exciting books also. But I think his most important contribution to science in Mexico, at least, is that he has built what I would say is the most important school of evolutionary biologists in our country. And that's easily said, but not done. Thank you very much. Welcome.
Thank you. Muchas gracias. Well, first of all, thank you very much for the invitation to be here with you. Thank you so much. I want to thank especially my very dear friend, Dr. Alfredo Herrera, for his very kind introduction. And one point that I would like to emphasize is that I'm not a physician, I'm not a virologist, I'm not an infectologist. The closest I am to medical sciences is my very professional attitude of a hypochondriac. But other than that, my main academic interest in the issue of the evolution of viruses is the fact that I'm very much engaged in the study of the origins of life. And when you study the origins of life, one of the key concepts that has been developing over the last 70 years, I could say, is the idea that there was, an RNA world, which we now recognize as a very early stage perhaps in the origin, but for sure at the early evolution of life. Now, let me just give a very, very brief introduction of what we understand today as the RNA world. And let me start by saying that in 1953, we had two major discoveries that were published with only 3 weeks in difference. First of all, the double helix DNA model was proposed by Watson and Crick in 1953 in Nature, in April. And a few weeks later, Stanley Miller was able to demonstrate that if you try to simulate the conditions of the primitive Earth under laboratory conditions, having an atmosphere with no oxygen, having an atmosphere with CO2, with methane, ammonia, hydrogen, water vapor, it was very, very easy. It only took a few hours, in fact, to synthesize amino acids, hydroxy acids, and some compounds that we believe may have been present on the primitive Earth. Now, what many people are not aware of is that 1 year after these two major discoveries were made, there was the founding of the RNA Club, which was an idea of George Gamow, a physicist, who came up with basically useless models of the evolution of the genetic code, but he was a driving force in trying to open the mind of people to theoretical issues in biology, and by James Watson. And the RNA Club, the RNA Club was, the RNA Club was an effort to put together a number of people engaged in RNA itself, in the relationship between RNA and the genetic code. And they used to meet basically at the so-called Cosmos Club in Washington, D.C. I have never been there myself, but it was chosen because the whiskey was supposed to be very good and very inexpensive. So there were good scientific reasons, if you wish, to meet there. Well, in any case, the founding of the RNA Club is an indication of how keen the people were from the very beginning on the relationship of RNA to the early evolution of life. Now, as time went by, in the 1960s, many people became fully aware that protein synthesis was possible in the absence of DNA. You only require a ribosome, in which, as Francis could say, there was an extraordinary abundance of RNA and unexplained abundance of RNA molecules. There were, of course, transfer RNAs, and there was the messenger RNA. And this was enough to have a synthesis of protein. So people began wondering if life was possible in the absence of DNA. And then in the 1960s also came what I think is an extraordinary demonstration of scientific intuition. Independently, both Leslie Orgel and Francis Crick came to the conclusion that if you look at the three major classes of molecules, which they were very keen at the time, DNA is a fantastic molecule, of course, with the double helix structure that explains replication, that explains inheritance of genetic characters and so on. But if you compare the relatively boring structure of DNA with the very complex three-dimensional structure of proteins and of RNA, then it could be predicted that perhaps RNA itself also had catalytic activity. And this was an extraordinary prediction that led, on the one hand, to the idea of the so-called Rna World Hypothesis that was proposed independently by Crick and by Leslie Orgel. Although their offices were side by side, they decided to write the papers independently. And the idea was that in the primitive oceans, prior to the emergence of proteins, prior to the emergence of DNA, there was RNA that was able to replicate, there was encoding for catalytic RNA, and that it was helping to set up a cycle that became known as the RNA world. Now, the RNA world hypothesis and the origins of life is a very coherent, but not necessarily seamless evolutionary narrative. And we believe that what happened was in the primitive Earth, there was a synthesis and accumulation of organic compounds, somehow by unknown processes. And this is a very open discussion. This led to the RNA world. And in the RNA world was where protein synthesis was invented. This idea is strongly supported by the fact that in the sanctum sanctorum of the ribosome, in the active place of the ribosome, we see no proteins, and that's precisely where the peptide bond is formed. So this strongly suggests that indeed protein biosynthesis was invented in the RNA world. The question marks that you see reveal the many unknowns that we have when we are trying to understand the origins of life. But specifically in this drawing, the idea is that we are not sure how far back in time we can extrapolate to the previous stage, but we are deriving for the current knowledge of biology. Now, we have no evidence whatsoever, direct evidence, fossil evidence. We have found no organisms that are actually made of RNA and protein. But the idea is that we can get some insights into the nature of biological systems prior to the emergence of DNA that can be provided by prebiotic model synthesis. We can try to synthesize the bases, the sugars under laboratory conditions. We know that we can find in meteorites there, as well as the solar system. We do find that ribose derivatives, we find bases without any problem. We can do the evolutionary studies of RNA populations, so-called select studies of RNA populations, and this gives some idea of the nature of the Darwinian processes acting over RNA populations. We can come up with theoretical models of RNA genetics, that is very frequently done, and of course we have RNA viruses. Now, it can be argued that some of the components of RNA viruses are quite old. It's quite unlikely that the RNA viruses themselves are direct descendants of the primitive RNA-based life. I can give you a number of arguments, but I will not indulge in that. We can discuss it later on. Nevertheless, RNA viruses can provide, even if they are not direct descendants of the first RNA life forms, they can provide direct insights into the structure and the evolution of early cellular genomes prior to the emergence of DNA. And the insights can give some information on genome size and organization, on replication strategies, on mutation and evolutionary rates. People tend to confuse the two, but they are clearly not the same. And gene generation and accretion processes. How do RNA genomes grow? Let me just remind you of the extraordinary fact pointed out by David Baltimore over 50 years ago, that if we look at the many different strategies in which RNA viruses organize its genomes. Compared to DNA viruses, this is clearly telling us that RNA viruses have explored an evolutionary space that's wider by far than that of DNA viruses. They have really been able to profit from the absence of competition of DNA viruses or DNA-based cells in a space that has allowed many different strategies, which is quite extraordinary. But one point some points that all of them share is that due to the lack of proofreading and repair mechanisms, most RNA virus genomes have error-prone replication. We know, of course, that they have very high mutation rates compared to DNA-based systems. They have extensive genetic variation, which of course becomes a public health issue, like in the problems that we have trying to derive, for instance, vaccines for a number of RNA viruses, like the HIV virus. They tend to have small sizes. They have different genome organization strategies. Some RNA viral genomes can be fragmented, as in the case of influenza. Some of them can have double RNA molecules, small double RNA molecules. Some of them, like the SARS or the coronaviruses, have these huge, large RNA genomes. And of course, they are restricted by a number of adaptive constraints that are derived from the properties of RNA. Now, what is very surprising, and we really do not have, no single person can provide you with a reasonable explanation of why the only common feature shared by all known RNA viruses is the monophyletic right-hand monomeric RNA-dependent RNA polymerase. In other ways, all the different RNA viruses do have the same type of polymerase. And clearly, the 3D structure of this polymerase is an indication of the monophyletic origin of this polymerase. And this makes, of course, the polymerase a natural target against viral infections as a unique phylogenetic marker. Let me concentrate a little bit on the first fact, the fact that the RNA polymerase is a natural target against viral infections. When we look at the 3D structure of, for instance, the hepatitis C virus RNA polymerase, we see that it's basically the same right-hand structure that we see, for instance, in the SARS-CoV-1 and CoV-2 polymerase without any problem. And that led very quickly in the 1920s, in the past 2 years ago, that led a number of people independently to suggest that perhaps sofosbuvir, actually the name is not shown here, but sofosbuvir, which is a medication that has been approved by the FDA that is known to have no secondary effect in the hepatitis C patients, could perhaps be used as an inhibitor of the SARS-CoV-2 virus replication. And independently, with actually few days of difference, different groups, including our own, published a paper suggesting that sofosbuvir could perhaps be used to redirect to the treatment of SARS-CoV-2, of COVID-19. It's easier to say than to be done, but a number of clinical trials are still actually going on. We ourselves are preparing, in the finished stages of preparing a report of like a clinical trial done in the Instituto de Cardiología in Mexico City, thanks to the initiative of our physician colleagues. And we hope to submit the paper very soon. And the results are encouraging, but as you know well, we still lack a definitive medication to treat SARS-CoV-2, especially solely by inhibiting the replication. Now, let me turn to the issue to which I feel much more comfortable, which is the fact that, in fact, the RNA polymerase is a unique phylogenetic marker. And let me just remind you briefly of something that I know that I'm sure that all of you know quite well, namely that the reconstruction of the evolutionary history of RNA viruses is hindered by the high level of mutation, which rapidly erodes the information contained in their sequences. So we have decided, since a few years ago, a number of groups independently came up to the conclusion, so why not use the 3D structure of the molecules to make phylogenies? And in fact, this is possible because most polymerases are part of a monophyletic group, and they lack absolute substrate and template specificities. You see here the RNA polymerase of SARS-CoV-2, of the reverse transcriptase of HI1, and a number of DNA polymerases from E. coli, the family B, the family Y, and the family A. And clearly the three-dimensional structure shows that they are monophyletic in origin. Well, we have just submitted a few days ago a paper that is actually an update of a paper we published in 2050, showing a phylogeny based on the 3D structure of the molecule. Here you see the polymerase, the family Y of DNA dependent DNA polymerases. Here you see all the reverse transcriptases, and here you see the different families of DNA and RNA viruses all together. And the fact that they form very clear groups, they are consistent with the biology of the viruses, which is of course an indication that our tree is not so wrong. And now one thing that I have to emphasize is that due to the absence of editing and repair mechanisms, RNA viral genomes are constrained by the so-called Eigens Limit, which is actually a wonderful demonstration of the interaction between theoreticians and laboratory studies. And they tend to have very small sizes, the RNA viruses. The only exception are the so-called nidovirales, of which include the coronaviruses, because they have 3' 5' exonuclease. And this is a very distinct characteristic of the coronaviruses, which of course allows, as you can see in this wonderful graph, actually this one is shown better, where you see the relatively small sizes of the genomes of RNA viruses, with the exception of the coronaviruses. And this is due, of course, because if you have something that's editing the product after replication, then of course the product, the molecule can increase its size. Well, this led to the question of what is the ultimate origin of the exonuclease that we see in coronaviruses. And in August 2019, a few months prior to the pandemic, a rather bright undergraduate student joined the lab, Adrian Cruz Gonzalez, and he began working with Rodrigo Jacome, my former PhD student, now my colleague, also part of the lab. And we decided again to go to 3D comparisons instead of sequence comparisons. And here you see the fantastic level of identity between the SARS-CoV, this is the one, but they're basically the same, and the E. coli shown in red. You see how we can superimpose the structure, and we have absolutely no doubt of their homology. We have developed techniques, I will be more than happy to discuss them afterwards with you, of comparing, of trying to find out the different structures that are homologous. And here you see the exonuclease domain of E. coli DNA pol 3. Here you have for the Lassa viruses, which is also an RNA virus. Here you have for the fake T7, a DNA virus, and for SARS-CoV-1. It's basically the same as the SARS-CoV-2. Well, again, we applied the method we had developed in the lab to come up with phylogenies based on three-dimensional structures. And here you see that three distinct lines indicate that at least 3 times viruses have hijacked exonuclease genes. One of course is the case of the double-stranded DNA viruses like the DNA fakes. The other case is the single-stranded negative RNA virus like the Rna Viruses, an independent hijacking of the exonuclease gene, and a third one by the coronaviruses, where you see very clearly separated. So what is this phylogeny based on 3D structures indicating to us? It's telling us that the exon and exonuclease genes have been hijacked by viruses at least 3 times by DNA fakes, Phi29, T4, T7, and twice by RNA viruses, once by Rna viruses and coronavirus, which of course have RNA genomes. In other words, and this is typical of many enzymes associated with DNA and with RNA, exonucleases activity includes both the RNA and DNA. Like other nucleic acid associated enzymes, they lack absolute template and substrate specificity, an issue that was actually predicted in the 1970s by Emil Zuckerkandl, the founder of molecular evolutionary studies. So again, from the perspective of the origins and early evolution of life, I think that it's very reasonable to assume that the early evolution of an exonuclease represented a critical step in enhancing the encoding abilities of primitive RNA genomes. It may not be so difficult to evolve an exonuclease activity. After all, an exonuclease is simply a hydrolase reaction capable of destroying a phosphodiester bond in a polymer that is already strained due to the mismatch of the base pair. And the postulated transition from small fragmented RNA genomes to a much larger DNA cellular genomes would have been facilitated by the lack of absolute substrate specificities of exonuclease, which I already shown you that is there. Now, in coming to an end, if you look at any cellular genome nowadays, it's very clear that gene duplications have played a key role in enhancing the size, the encoding abilities of cellular genomes. However, in viral RNA genomes, multigen families are conspicuously absent, suggesting that gene duplications may occur, but their preservation is not favored. And of course, we have to keep in mind that the high level of mutation rates rapidly erodes evolutionary information containing sequences. The same is not true of the 3D structures. And so in order to test this, with one of my former students, Alejandro Martinez Cisneros, he did his master studies with me and now has moved to do his PhD studies with my friend Valeria Sousa, which many of you know. We decided to look carefully at the genomes of different RNA viruses. And this is an example of the, I do not know the name, BC stroviridae, which is a single-stranded RNA positive virus. And we look at the BPI1, BP2, and BP3 proteins, which are in the surface protein of the virus. And here you see the comparison. It was actually, I don't know why it doesn't appear here, but it doesn't matter. Here you can see the paper was published last year. And here you can see the comparison between these, the 3D structure of these proteins, a comparison that if you are using 3D can be seen quite clearly. The homologous origin of these duplications is clearly visible. But if you go to the sequences, you only see something like 12 or 13% percentage of identity. And we actually decided to look at another peculiar domain, which is the SARS unique domain, the SUD, which is here, which we know interacts with a human factor and enhances viral RNA translation. This is a phenomenon that has been very well described. And it turns out that SUD had been studied in the 2009 by a group, Tan et al. And they actually indeed suggested the homology of the domains of this protein. They actually superimposed the structures, but they didn't do it with a phylogenetic idea in mind, which was perfect because it became a control for our own methodology. And here you can see in this figure over here in the bottom figure, how the 3D alignment between SUD-M, which is this, and the X-domain cyan, which is here, shows that clearly there is a duplication, a gene duplication. So in conclusion, viral RNA polymerases and the E. coli DNA pol 1, 2, and 4 may be vestiges of the ancestral proteinic polymerase from the RNA protein world stage. This is an idea which I'm very keen on, and the question is, why this polymerase and why not the others? Secondly, the gene duplications or recombination may have played a key role in the size increase of cellular RNA genomes. And thirdly, a key event in the evolution of genomes, cellular genomes, must have been the emergence of an array of editing and repair mechanisms. And we posit that a very early one was precisely the appearance of the 3', 5' exonuclease activity. And I have to confess, I'm not sure all my co-authors will agree with me on this. They will, because they work for me in the lab. But I'm very tempted to believe that the polymerase and the exonuclease activity that we are seeing nowadays are actually remnants of a very, very early evolution of these molecules during the early stages of biological evolution. Thank you so much.
Thank you, Toño. Are there any questions for, Marco?
Thank you. Great presentation, Dr. Antonio. Did you include in all these genomes only viral genomes from humans, or you included from other animals as well, like
animals and plants, everything, every
plants
molecule.
as well, and plants follow the same pattern as well.
Yeah, for all extant biological kingdoms, if you wish.
Okay, thank you.
But of course, if you compare the DNA polymerase, let us say from animals or from plants, they have additional domains that are binded to the DNA polymerase. But in the case they have an RNA polymerase, like in the telomerase, it's included there.
Very nice talk. Can you predict from the protein polymerases the potential structure of the RNA polymerase made of RNA?
That's a major issue. The first one to suggest that this might be possible was actually Tom Seitz, to whom we own the first proposal of the mechanisms of DNA polymerization and RNA polymerization. And he wrote in the 1990, more or less, a short review in the Journal of Biological Chemistry. And he actually said that one could come out with a scheme in which different domains of a RNA polymerase, a ribosimic RNA polymerase, perhaps we could see how the substitution of small domains by a proteinic RNA polymerase could explain the transition. I have only once tried to do that. I spent about 3 weeks trying to see how it could be done, and my results were quite negative. I think that the answer there probably lies in the presence of a protein cofactor that was assisting a ribosimic RNA polymerase, and eventually the substitution came. But the real question would be not to bind the substrate or not to bind the template, but rather to have the actual catalytic mechanism involving the DD domain in the case of the polymerases, the zinc that partakes in the processes, and so on. It's a major issue.
Anybody else has a question? There's something a little bit unrelated to what you just told us, but the evolution of SARS-CoV-2 regarding also these recombination events, because we are now talking about this Delta Cron, I mean, a terrible name, but how is this influencing this pandemic or the evolution of the virus itself, and what can we expect in the future from this type of recombination? Is going to be a major factor? What is the destiny of this virus, in other words?
Well, nobody engaged in evolutionary studies will dare to make a prediction of the future. It's very difficult to predict the future, you know. But most of the proposals that I have seen have concentrated on the idea of the virus changing due to mutations, deletions, or additions, basically. And mostly the British group has concentrated on emphasizing the role of recombination. I think that we are bound to see much more events of recombination if we look for them. And don't forget that recombination will be very easy to detect if it's between very different sublineages or well-separated sublineages. It may be, actually, I have a student that proposed me the project and we try a little bit and it was not very encouraging to see, for instance, between subvariants of closely related subvariants, let us say, of Omicron or Delta and so on, but it's not so easy to detect the recombination there. It's clearly taking place. I think it's something we have to be very keen on. It's something that the exonuclease will never detect. The exonuclease will just go on repairing the mismatch nucleotide. And I'm not scared about our ability to detect recombinants, and nor worry too much as long as the recombinants includes especially the part encoding for the spike protein, because then all the vaccines and medications and so on, we have to inhibit that part will be working. But
Kat has a question, so I don't know.
Hi, can you hear me okay? Can you hear me okay now?
Hi, Kat.
yeah. Yes, that would be lovely, Kat, please. On the chat. Lo va a poner en el chat. Yes, Kat, please go ahead.
Sorry, I just very quickly typed my question in the chat, but can you hear me now? And now I can't hear you.
The availability of the cryocrystallization. Now we can actually have crystals in a very short time. So I think the combination of these different methods, Kat, will be extremely useful. By the way, sometimes we complain about the fact that Zoom doesn't work well, that the communication fails. May I advise you to read the extraordinary novel by Catherine Porter, Pale Rider, White Horse, that was written when she caught influenza in 1918. And the fact how isolated the patients, the physicians felt compared to a little bit of noise that we have to tolerate nowadays.
Thank you very much, Daniel, for
Thank
this
you.
wonderful talk.